So what is this genetic mapping? So genetic mapping is very important to find out various genetic disorders in human beings. Only the repetitive DNAs can be isolated from it. So for that, what they do is they digest the DNA, and the bulk DNA, which forms a major peak and other small peaks, are referred to as the satellite DNAs. Hello everyone, a warm welcome to the last session of chapter 6 of second PUC, that is molecular basis of inheritance. I'm Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the Human Genome Project, what were the goals of the Human Genome Project and why Human Genome Project was important and all that. In today's session, we shall study about a very interesting topic called as DNA fingerprinting and the techniques that are involved behind DNA fingerprinting. So to begin with, let us start with studying about DNA fingerprinting, a small introduction to it. So DNA fingerprinting is nothing but it involves the identification of differences in some specific regions in a sequences called as repetitive DNA. So, say for example, this DNA fingerprinting is of great importance during a crime, that is for the forensic team. Why? Because each person's DNA at some point they tend to vary. They are made up of the same type of genes, but in spite of that, because of the presence of tandem repeats in them or because of the presence of repeated sequence in them, having the same repetition of the genetic code based on the number of repetitions are there, that will vary from person to person. So based on that, whoever has done the crime, by identifying that person's DNA sample and sampling it and sequencing it, the culprit can be found out through either his hair or any skin tissue or through his nail or any small part that is available of him. So that is why DNA fingerprinting is very important. So it mainly involves identifying the differences that are there in some specific regions of the DNA, which is called as repetitive DNA. So what differences are occurring in the repetitive DNA strands of the DNA, that is found out in DNA fingerprinting. And these sequences, that is the repetitive DNA sequences, they are a small stretch of DNA that is repeated many times. And I told you, these repetitions are the ones that vary from one person to another. So these repetitive sequences, one, they have got a material. Say for example, they have got a nail. So that small nail material that is there, they will bring it to the laboratory. They'll separate the DNA out of it. So they'll separate the repetitive DNA from the bulk genomic DNA. So we all know that the human genome is about it is a stretch of DNA of several base pairs, right? In that stretch of DNA, some of the strands of the DNA are repeated multiple times. So which are called as the repetitive sequences. So only from that stretch of DNA that has been isolated, they will again isolate only the repetitive sequences that are there on that stretch of DNA, and that is by using different gradient centrifugation. So what happens, if you remember in one of the sessions we had studied, right, how that is DNA is semi-conservative in nature. There, how they had used the centrifugation procedure in order to separate the DNAs of different densities, right? So that is how, because each strand of DNA will have, or small fragments of DNA will have its own densities. Now, likewise, the repetitive DNAs can be separated based on their densities by the density gradient centrifugation method. So what is the centrifugation? How do they do it? So what did I tell you? First, they'll collect the sample. They'll try to isolate the repetitive DNA that is there in the DNA. After isolating the repetitive DNA sequence, now they need to, because there are a lot of repetitive DNA sequence in a strand. So now they need to separate each of these repetitive DNA sequences. So what they'll do is that sample, they would have taken it in a vial or in a centrifugation tube, which is a plastic tube. They'll take it inside that and then they will put it in a centrifugation machine. Inside that machine, what will happen is that machine will have different speeds of rotations. So when it rotates, because of the speed of the rotation, what will happen? The sample will get separated based on its weight or based on its density. So that is how the repetitive DNA is being separated here based on its density gradient using a density gradient centrifugation. And the bulk DNA, which forms a major peak and other small peaks, are referred to as the satellite DNA. So the bulk DNA. So apart from the repetitive sequences, the bulk DNA that is there, that will form the satellite DNA. So next we shall study about how satellite DNA is involved in DNA fingerprinting technique. So satellite DNA, depending on the base composition. So what is satellite DNA? After removing the repetitive DNA, what bulk DNA is left over, that will be the satellite DNA. So that satellite DNA is rich in adenine, it is rich in thymine, or it will be rich in guanine and cytosine. Why is it rich in adenine and thymine? Of course, if adenine is there, it will be rich in thymine because they are complementary base pairs. Two strands will be there, no? So that is why. So if they're rich in guanine, they'll be rich in cytosine, and vice versa like that. If they're rich in thymine, they'll be rich in adenine, and rich in cytosine means rich in guanine. So therefore, they are adenine, thymine, guanosine and cytosine rich. And the length and number of the repetitive units, which is the satellite DNA, is classified into many categories. So there are different types of satellite DNAs based on their length. So they are called as the microsatellites, the mini satellites, etc. So these microsatellites, that is satellite DNAs that are there, these sequences, they do not code for any of the proteins, means they are the non-coding sequences, but they form a large portion of the human genome. That is true, right? Because human genome means human beings are eukaryotes. So eukaryotes means they have the DNA, and in that DNA, both the coding strands and the non-coding sequences, the coding and the non-coding sequences, that is the exons and introns, both are present. Now, the number of introns when compared to exons are more. So therefore, here, what happens here is, what are these satellite DNAs? So satellite DNAs are nothing but the repetitive DNAs that are present on the DNA strand. And these satellite DNAs, that is based on the number of repetitive units they are made up of, or based on the number of lengths they are classified as microsatellites and mini satellites. And these microsatellites and mini satellites, which are nothing but again sequences, they do not have genes which code for a particular protein, means they are non-coding sequences. They do not participate in the formation of a protein. So therefore, these form a large portion of the human genome. And these sequences, they show high degree of polymorphism. Polymorphisms means change. They are capable of changing. They show high degree of polymorphism and forms the basis of DNA fingerprinting. Why do they have a high degree of polymorphism? Because I told you repetitive DNAs, they vary from one person to another. They are not the same in every person. That is why, though the coding genes that are there, usually in all the people, the coding genes will be the same. Why? Because they have to produce the same kind of proteins, right? Otherwise, what would have happened? In human beings itself, different species would have existed. So that is why in coding sequences, almost all the coding sequences are similar in all the human beings. But when it comes to non-coding sequences, which are the repetitive DNAs or which are the satellite DNAs, they tend to vary from person to person. Based on that particular variation, the culprit can be found out. So that is how DNA fingerprinting. So therefore, satellite DNA is the one which is a non-coding sequence, repetitive sequence. They are the ones which actually forms the basis for DNA fingerprinting. So now we know what is repetitive DNA. We understood that what is satellite DNA, and we came to know that those sequences, that is the repetitive, present in the repetitive DNA, they are non-coding sequences, and they are therefore and they vary from one person to another. So therefore, they are used in DNA fingerprinting. So much we have understood. So moving further, so here, since DNA from every tissue, from where and all can the DNA be isolated? Because what do they do first to find out they first have to get a material which is containing the DNA, be it a small portion of the cell, any cell, right? So from where and all can the DNA of a person be isolated? It can be isolated from blood. So maybe somewhere there might have been a fight between two individuals. So one of the individual might have killed that person, and during that fight, this individual's blood would have spilled out during the fight. So therefore, taking that blood also, the forensic people can isolate DNA from that, and they can identify who the culprit is. So by blood, through hair follicles, through skin, through bone, through the saliva, sperm, etc. Even a small portion of the skin, if it is peeled off, any cell they get, they can isolate the DNA out of it. So from an individual, all that is taken and they show same degree of polymorphism. They show difference in the repetitive DNAs. So that is why it is of great importance. And this polymorphism is the one that actually becomes very useful tool in identification in forensic application. So that is why the crime reporters, or not the crime reporters, people, the detectives or the forensic people and all that, they find out the culprits based on this particular DNA fingerprinting technique using isolating the DNA from blood, hair follicles, skin, bone, etc. So further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity as well. But this polymorphism, these repetitive sequences that are there, can be inherited from the parents. Say for example, there is an orphan kid. Somewhere the kid has been lost at a very young age, when it is right from the hospital. Now will the kid know who its parents is? At that time also the DNA of the kid can be isolated, and any parent saying that this is my particular kid, from that parent also the DNA sample can be isolated and they can be matched for the polymorphism or for the repetitive DNAs or the satellite DNAs that are present in that particular chromosome or the DNA strand. So based on that, as I told you, because these particular repetitive DNAs or this particular polymorphisms are adapted or it is transferred from the parents to the children as well, the DNA fingerprinting is the basis for paternity testing in case of dispute. So usually we know that this happens, right? Some baby goes missing from the hospital because if it is a boy baby, usually what they do, some other family members will just exchange it without the knowledge of anybody. So that is what happens sometimes, right? That is the time when to find out who the exact parents is, this particular DNA fingerprinting technique can also be followed. And polymorphism in DNA sequences is one of the basic requirement of genetic mapping. So what is this genetic mapping? So genetic mapping is very, very important to find out various genetic disorders in human beings. Like we have, say for example, Down syndrome, right? So there are babies or children who have Down syndrome, but can the Down syndrome be found out earlier itself? It can be found out. How can it be found out? It can be found out by chromosomal mapping. So here what happens in the case of Down syndrome is, usually we human beings, we have 46 chromosomes, right? That is 23 pairs of chromosome. But in a Down syndrome person, an extra chromosome will be in the 21st chromosomal pair, leading to a trisomy condition. After genetic mapping is done, when they find out that there is an extra chromosome on the 21st chromosome, they'll find out that, okay, this baby might develop a Down syndrome or it is having a Down syndrome. So therefore, genetic mapping is very, very important. And for that genetic mapping also, DNA polymorphism in DNA sequences is one of the bases for genetic mapping of human genome, as well as it is the basis for DNA fingerprinting as well. So this is about the DNA fingerprinting. So far we had learnt about DNA polymorphism. Everywhere I had used the term DNA polymorphism, right? So what is it? Let us look into. So if an inheritable mutation is observed in a population at a very high frequency, it is referred to as DNA polymorphism. What do you mean by polymorphism? Differences, right? We are all polymorphic. to say, of course, we are all polymorphic, right? We are all different from each other. So that is what sometimes what happens, DNA tends to mutate, DNA tends to change. And I told you the repeated DNAs tend to vary from one person to another, leading to DNA polymorphism. So these mutations will keep on getting accumulated. So mutations means it is any change that occurs in the DNA strand is nothing but called as mutation. And that mutation will bring about polymorphism. Maybe it will change the skin color of the person, the hair color of the person. It can cause a lot of deformities and all that, right? So that is why these mutations, what happens from generation, say for example, there are two parents, healthy parents, but the son or the daughter that is there will, because of DNA mutation, she might develop some patches in the skin. Say for example, for you to understand, some brown patches in the skin. Maybe that progeny's child will also have that brown patch. Next generation also they'll develop the brown patches in the skin. So that will be carried from generation to generation. So these mutations later on what will happen, it will keep on accumulating generation after generation, and from one of the bases of variability and it will form one of the bases of variability or polymorphism. So that is why each individual is polymorphic in nature because of the changes occurring on the DNA strand or the mutations that occur on the DNA strand. So there is a variety of different types of polymorphism ranging from a change just in the single nucleotide or very large scale changes. If you remember previous session, we had dealt with DNA mutation, right, wherein we studied about mutation of a single nucleotide, which is called as point mutation, and we studied about frame shift mutation, wherein frame shift insertion mutation, frame shift deletion mutation. So go back to the previous session and have a look at it. You will understand very well. So therefore, these mutations or these changes in a nucleotide brings about the polymorphism. So that mutation will be carried on to the next generation. Likewise, it will get carried throughout the generation, leading to a different set of individuals having different characters or different traits. So for evolution and speciation, therefore polymorphism is very, very important. And it is because of this polymorphism nature of the DNA itself, that is why it is also of great importance in DNA fingerprinting in the forensic science. So therefore, this is about DNA polymorphism. And the word itself, it says polymorphism means many changes occur in an individual because of mutations occurring in the DNA or because of changes occurring in the DNA strand, either because of change in one particular nucleotide or a small stretch of the nucleotide. So that is about DNA polymorphism. So moving further, let us study about DNA fingerprinting technique. What is done in DNA fingerprinting? How is it exactly done? So this technique of DNA fingerprinting was actually invented by a scientist, Alec Jeffreys. So what he did was, what is used in DNA fingerprinting? We learned in the previous slides, it is the satellite DNAs, right, which are nothing but the DNAs which do not code for any proteins. They are non-coding in nature. They do not produce any proteins. So what he did was, he used the satellite DNA as a probe, and this satellite DNA that he chose had high degree of polymorphism. Now, you know what polymorphism is, you know what satellite DNA is, so I need not have to keep repeating to you again and again. That is why earlier itself I explained to you what satellite DNA is and what polymorphism is. And it was called as variable number of tandem repeats. Why tandem repeats? Because they get repeated a number of times on the DNA strand. They are repetitive DNAs, right? So that is why. So, and these repetitive DNAs have a great variations. So that is why they are called as variable number of tandem repeats. So this technique was used earlier. It was using a method called as Southern Block hybridization using radio-labeled VNTRS as a probe. So first what they do is, they actually create a probe. That is, if you can see here, it has a tandem repeats. Number of tandem repeats are there. These tandem repeats in each of the chromosome or on the DNA strand, it has to be separated. So again, for separation, what they do, they go in for the gel electrophoresis and all that. And especially here they follow a method which is called as southern blotting. In southern blotting, what they do is they actually use a radioactive probe. So that probe is complementing to the repetitive DNA strand. It will, or to the particular satellite DNA strand, it will go and bind there. It will go and bind there and it will make it to fluoresce or emit certain color, therefore helping us to identify that particular strand. So that is how it is done. So there are different steps involved in DNA fingerprinting technique. What are those? We shall look into. The first step that is there, first we need a DNA strand, right? So for that we need to isolate the DNA. So first step is isolation of DNA. Now after isolating the DNA strand has to be broken down into smaller pieces so that only the repetitive DNAs can be isolated from it. So for that what they do is they digest the DNA. They digest the DNA by using an enzyme which is called as restriction endonucleases. So when they use the restriction endonucleases, what happens is that after that the separation of DNA fragments is done by gel electrophoresis. What is done in a gel electrophoresis is, first they would have isolated the DNA. That DNA they have to break it down into smaller fragments. For that they add a particular enzyme called as restriction endonucleases. Now they have got different fragments of the DNA. Now from that fragments of DNA they have to separate the repetitive DNAs and the other DNA strands. Everything should be separated. How will they separate it? That sample which was containing the DNA strands, right, that they will take it and they will load it into wells. So when they load it into wells, what happens? There is a gel-like thing. So there is a gel plate here. So on this plate, agarose gel will be added. So in that agarose gel, what they will do is there will be small wells or holes here. So to this, they will load the sample. When they load the sample and they pass electric current, what happens that is, in that electric field, when that electric field is created, the DNA strands get separated based on their size. So smaller the DNA strand will move very far. Heavier DNA strands will just move a little bit distance. So therefore now, based on the weight or the size of the DNA strands will get separated. So that is what they do. That is, they separate the DNA fragments by electrophoresis method. So in biotechnology principles also, this chapter, that chapter you will study this very well. And next, after that, now these separated strands that have been got, what they will do is they transfer it from, they'll remove it from this particular gel electrophoresis and they will transfer it into a blotting sheet of separated DNA fragments to a synthetic membrane. They'll transfer it to a synthetic membrane such as the nitrocellulose membrane or the nylon membrane. From where did they transfer it? Now here the fragments of DNA had got separated, right? Those separated fragments of DNA, they'll transfer it from here to one more membrane. This is a membrane which is made up of nitrocellulose or it can be a nylon membrane. To that they will transfer it. Now after transferring, they will go in for hybridization using labeled VNTR probe. So variable number of tandem repeats, that is what is used in southern blotting, right? Because here southern blotting technique is followed. So next what they will do is they will go in for hybridization method, wherein they have to find out in this separated DNA which are the tandem repeats that are there, which are the repetitive sequences that are there, that they need to find out. So for that particular variable number of tandem repeat probe would have been designed already. So that probe they will introduce it here on this nitrocellulose plate, and later on the detection of hybridization DNA fragments by autoradiography will be done. So when autoradiography, what it will do is it will start to emit color showing that where these probes have actually gone and bound, and that particular probe we can separate, we can sequence it, and then we can do the DNA fingerprinting. So that is how DNA fingerprinting technique is done here. It is a very beautiful process which in your PUC they have just given it in small steps, but as you go to your higher classes you will study a lot of it. So biotechnology, those who are very interested in studying about DNA, about genetic mapping, the fingerprinting and all that can take up biotechnology, which is a quite quite very interesting subject, and also it has a lot of scope as well. So next, so far we were talking about VNTRs. What are these VNTRs exactly? They are the variable number of tandem repeats, right? So VNTRs belong to a group of satellite DNA, which is referred to as mini satellite. I told you satellite DNAs are of different types. They can be mini satellites, they can be macro satellites, micro satellites, and all that, right? So these VNTRs, they are the satellite DNAs which belong to the mini satellite. They are called as the mini satellites. And small DNA sequence is arranged randomly in many copy numbers. That is true, right? Because they are repetitive in nature. On the DNA strand, they are arranged in many copy numbers. And the copy number varies from one chromosome to another in each individual. Because of this variation itself, it is one of the best chosen segment of DNA for DNA fingerprinting. And the number of repeats shows very high degree of polymorphism, and as a result, the size of the variable number of tandem repeats varies from 0.1 to 20 kilobases. So the size varies because it keeps on changing from one individual to another. So that is why, and from one chromosome to another, the size of the VNTR or the number of repeats of the VNTR tend to vary. So this is about the VNTR, which is the variable number of tandem repeats. So next, let's look into the DNA fingerprinting application. So I told you about southern blotting and all that, right? It is very vast to study about southern blotting, but in your PUC, now at least we have given you the basic of what is southern blotting. So what is done in southern blotting? From gel electrophoresis, whatever DNA strands, they were separated, DNA fragments were separated, that will be transferred to a nitrocellulose or a nylon membrane. On the nylon membrane, to those strands, special variable number of tandem repeat probes will be designed. Those probes will be put into that. So those probes will be designed in such a way that they are complementary to the tandem repeats. They'll go and bind there. When they go and bind there, after that, they'll have a particular color given to that, which will fluoresce when it is radio labeled. So that is when based on the color, we can find out, okay, these are the tandem repeats. Those tandem repeats can be isolated. Those tandem repeats will be put into a sequence analyzer. And in Human Genome Project, we had studied, right, how in the computer, the sequence were shown, right, A, T, G, C, like that sequence, that will come. So that is when, so if one of the tandem repeat sequence is ATGC, then from the crime scene, the blood sample that is collected, when they check that, that will also be having the tandem repeat of ATGC itself. Therefore, it is understood, okay, this is the person who has made that particular crime. So that is how DNA fingerprinting is done. It is very interesting. You can read a lot about it if you have great interest. So this is about DNA fingerprinting application. So where all it is used. So DNA fingerprinting is used in forensic science to find out the culprits or the criminals, and they are used to determine the population density. Why? Because in each individual, the polymorphism exists because of variation in the that is the variation in the tandem repeat. So that is why they can be used in determining the population diversities, and they also can be used in determining the genetic diversity. Genetic diversity and all that, why it will occur? Because I told you DNA strands at some places tend to mutate. That mutation will also result in polymorphism. Therefore, genetic variation will keep on occurring. So for that also DNA fingerprinting can be used. So this was about the DNA fingerprinting technique. So in this particular session, we learnt about a DNA fingerprinting. In DNA fingerprinting, we learned what satellite DNAs are. We learned what variable number of tandem repeats are. Then we learned how exactly the different steps that are involved in DNA fingerprinting, and all that we had studied. And also we came to know about few of the important applications of DNA fingerprinting in the field of science. So with this, the chapter comes to an end. So I hope you understood the chapter very well. So we shall meet again in the coming sessions with a new chapter. So see you then. Thank you.